And it is my great pleasure to introduce our second speaker for today. Francis Barany is a professor of microbiology at the Weill Cornell Medicine. Barany is the best known for inventing the ligase chain reaction, ligase detection reaction, and the universal DNA arrays, which are the foundations of the commercial test to diagnose the genetic disease, detection of infection pathogens, and profile cancers using the DNA microarray and targeted next generation sequencing. Barany holds 74 issued USA patents and over 100 international patents that are widely used by molecular diagnostic and sequencing companies. Thank you very much for joining us, and thank you very much for focusing also on targeted protein degradation today. We are very excited and very much looking forward to your seminar.
Thank you, Mikolai. I really appreciate the kind introduction. I'm just going to share my screen, and okay. Here we go. So today I'd like to talk to you about CurePro's reversible self-assembling drugs for targeted protein degradation. But I also would like to give a shout out to the Dana-Farber Cancer Institute Targeted Protein Degradation webinar series, which I have been following since its inception. And there were many Thursdays that was the highlight of my day during the pandemic. Here's the obligatory conflict of interest slide. And I want to bring you back over 50 years ago in 1971, when then President Nixon declared the war on cancer. Now, at that time, over 330,000 people in the US died of cancer that year. If we fast forward some 50 years later, in 2024, there are over 2 million new cases of cancer, and 611,000 cancer deaths are projected. Almost twice as many people will die of cancer than 50 years ago, but our population has not doubled during that time. So why is that? Well, current treatments are based on burning with radiation and poisoning with chemotherapy, which ironically often actually accelerate metastatic spread. And while we all believe in precision medicine, it unfortunately helps less than 10% of cancer patients, as tumors inevitably undergo mutational escape. So if we fast forward to the mid-2000s, I was director of an NCI-funded program project grant collaboration between Memorial Sloan Kettering, Rockefeller University, Princeton University, and Weill Cornell Medicine, where we were doing the molecular profiling of colon tumors. We were looking at expression levels, at copy number, mutation, methylation, which was really a forerunner to the current cancer genome anatomy program. And there were two things that came up from this work. The first was that changes in gene expression level were mirroring changes in gene copy number, and that oftentimes gains of entire chromosomal regions were driving changes in expression, for example, the 20q region that is over amplified in many different tumors. Now, here is an example of a gene we all know and love, MYC, which plays a major role in over 50% of all cancers. And what we see here is expression level in standard deviations, the lung, normal tissue, normal liver tissue. And then we see in blue, the normal colonic tissue, and polyps shown in green. You see 2 standard deviation shift to a higher expression. We also see the same in tumors and in metastases. If we now look at copy number, the normal copy number is 2, but in both tumors and metastases, we see quite an increase in copy number. And if we display both pieces of information from the same tissue samples, we can see that expression can be driven by both increases in copy number in this direction, as well as increases in expression which are copy number independent. For example, if MYC expression is being driven by another protein, such as BRID4. And the second thing that we noticed was it wasn't just a few dozen oncogenes, as was in the classic textbooks of the time, but there were hundreds of upregulated genes, which are illustrated here. So blue means that the expression is down, and orange and red means expression is very high. And MYC was not even in the top 50 genes in terms of overexpression. Now, there are some genes here you might recognize, but there are many other genes which are transcription factors and scaffolding proteins, and proteins involved in protein-protein interactions, as we have just heard in this beautiful talk before ours. And what we see is that most cancer proteins are currently considered undruggable due to a lack of an adequate binding site. So at the time, we were thinking about how could one drug undruggable proteins, and we came up with the idea of cofuorons. Cofuorons use bioorthogonal reversible linkers to assemble larger drug molecules to then drug the undruggables. So in our scheme, we have two molecules which are covalently attached to each other in the pill. This is then ingested. And once it hits the stomach, which is in an acidic environment, the two molecules come apart. Each one comprises of a pharmacophore or ligand and a linker element. As small molecules, they are absorbed and distributed throughout the body. And then they can both enter into a cell. And once they are inside the cell, they can reassemble on the protein target and thus get activity. So here we see an example of the cofuoron effect. Here is a ligand for tryptase, which is appended to a catechol linker. Here is a ligand for or another tryptase ligand with phenylboronic acid. And when you bring these two together, you create this larger molecule. Now, each one of these alone has an IC50 in the sub-micromolar, but when you put them together, the IC50 shifts to 14.8 nanomolar, and this 40-fold improvement, we've termed the cofuoron effect. So here we can see in a three-dimensional plot, one, the other molecule, and then when you combine them, you see this dip in the mountain, and that indicates this cofuoron effect. Further, if you compare to IP injection against the vehicle control, we can see after 24 hours and 2 hours and 8 hours and 24 hours, we get a substantial reduction in tryptase activity, showing that these molecules come together in vivo to have biological activity. Now, meanwhile, as this audience very well knows, there were innovations that were occurring here at the Dana-Farber Cancer Institute, Jay Bradner's lab, and simultaneously in Craig Crew's lab at Yale, the development of proteins. Others done some very elegant work on molecular glues. And these generally are bifunctional molecules, which once they enter into the cell, they bring the E3 ligase machinery in proximity to the protein target, enabling ubiquitination and then allowing for proteasomal degradation. Now, after that degradation, the molecules release and can affect the next degradation. So this is event-driven mechanism of action. Yet, nevertheless, there are some drawbacks in that most degraders lack drug-like properties due to their large size. They generally only work by one mechanism, the event-driven mechanism. These molecules are slow and expensive to produce and optimize, and they are evaluated and optimized one at a time. So we thought we would bring together the best of cofuorons and PROTACs to create cure pros, which are reversibly self-assembling drugs. See, one adds the two monomers, which under physiological conditions, the equilibrium is 99% plus in the monomer form versus the dimer. As monomers, they readily traverse the cellular membrane, and once inside, they can again organize formation of a quaternary complex, which brings the E3 ligase machinery in proximity to the protein, enabling for proteasomal degradation of the protein target, liberating the two monomers, which can then recycle to affect the next degradation. Now, again, we do take advantage of the coferon effect, and we have shown that even with ligands that bind with poor affinity, when combined with the machinery, one can get ready protein degradation. Their advantages are quite clear. As small molecules, they abide by Lipinski's rule of 5, so they easily get into cells. There is a dual mechanism of action, namely, one of these molecules, the red molecule, can act as a traditional occupancy drug and inhibit the protein function, while the combination of both the blue and the red molecules allows for ready degradation, so it's an event-driven mechanism. Synthesis and optimization are fast and cost-effective, and the in vitro combinatorial screening operating system, which I will show you momentarily. So shown here are two cure pros. This one here uses the JQ1 BRID ligand developed by the Bradner lab, and which we've appended a phenylboronic acid linker. And this one here uses the VHL ligand developed by Zueli when he was in Craig Crew's lab. And here we've appended the catechol linker. As you can see, when you put these two molecules together, one can form this larger molecule as the reversible boronate ester formation, which forms a 5-membered ring. Now, one molecule alone, we see there's no degradation of BRID4. The other molecule alone, again, good levels of BRID4. But when you have both of them together, you can obliterate BRID4. Furthermore, it turns out there is no hook effect, because even at high concentrations, you get very, very good degradation. And here is a high-bit assay to show that that degradation is very rapid and sustained. Indeed, if cells are treated with the combination of both of them, you obliterate within the first 4 hours. That's sustained over 8 and 12 hours. However, if you treat for 4 hours and then wash away the excess drugs, after time, you start seeing rebound of the BRID4. Here we see our combinatorial matrix. So this is the first high throughput protein degrader discovery platform. Illustrated here are 10 BRID4 ligand monomers and 17 E3 ligase ligand monomers, 13 of them to cereblon, 2 to VHL, and 2 to MDM2. And this matrix immediately allows you to have 170 cure-pro pairs. Furthermore, these E3 ligase monomers will be redeployable when we go after our next target. What you can see immediately is out of the 170 cure-pro pairs tested, approximately a third of them degrade BRID4 greater than 50%. 22 degraded greater than 75%, and 4 degraded greater than 95%. So you can immediately find your best combinations to affect the highest level of degradation. It turns out that we can get degradation using multiple different E3 ligases, and it is very much dependent on the linker and the geometry of that linker. Shown here again is the BRID ligand with a VHL ligand, and we get excellent degradation. In the middle panel, we see a different linker with the BRID ligand, and here a cereblon ligand, and again, we get excellent degradation. And the lower panel, we see a BRID ligand. Now we've appended a catechol to the BRID ligand, and to the MDM2 ligand, we've appended the boronic acid moiety, and we can again see excellent degradation of BRID4. Now, I would like to draw your attention to this side of the slide, where we see that molecule, which is like 55, instead of having the hydroxyl at the ortho and meta position, if we just move it to the metal and the para position, as in number 54, we see that we've completely lost the ability to degrade BRID4. So just a simple moving of one hydroxyl from the para to the ortho position is the difference between no degradation and full degradation. Furthermore, when we look at the cereblon, we can see here that this 63 does not work with any of the cereblon linkers that are illustrated here. But if you use 65, then again, you can get good degradation. So once again, it is very dependent on which linker you're using and the geometry of the linkers. In addition to that, here we show the mechanism of action. We have two molecules which are almost identical, one which has the boronic acid linker and the other which just has a phenyl group. And what you can see is that each compound by itself, there is no degradation of BRID4. If you have no linker present, there's no degradation because the dimer cannot form. But if you have both the phenylboronic acid linker and the catechol linker, then you can get good dimer formation and degradation. Furthermore, if you now do competition experiments with either VH298, which is a VHL binder, or JQ1, which is a BRID4 binder, we can see that in either case, we do not get degradation. Only when we have the two pure-pro molecules present without any competitors do we get the full degradation. And finally, shown here, if we add the proteasome inhibitor, we can see that that completely reverses the proteasomal degradation, so that this shows that this is dependent on active proteasomal action. Just would like to briefly segue into the mechanism of action. Here we have these different reversible linkers, and we've determined what the K equilibrium of these different ones is. And then you can figure out what the monomer concentration is. And under physiological conditions, which is between 1 and 0.1 micromolar, we can see that less than 1% is in the dimer form. With our collaborators, Wei-Jin Gui and Tom Kodadek, we have shown that if you append a 2-acetylphenylboronic acid to a VHL ligand, and on the other VHL ligand, one appends an alkoxyamine linker, then if one tests various variations in linker length, we can see that one of these gives very nice degradation, although not at the same level as a dimer created in the Sueli lab, where you get significant degradation. Nevertheless, we can see that when we bring these two molecules together, you can reversibly create the covalent molecule that then has good activity against the VHL protein. Shown here, we can see that if we add these two molecules at different concentrations, ranging from 0 to 10 micromolar, the higher the concentrations of both of them, the greater the degradation. And furthermore, we can get excellent degradation even when one of the molecules is in larger excess compared to the other. If we now look at our PK and PD parameters, we can see that there's reasonable stability of these compounds, both in mouse and human. And when we look at the plasma concentration, we see that there's good enough AUC and T half-life for us to now proceed and go ahead with testing these in animal models. So here we see the cure pros again, 63 and 55. We then provided these to mice bearing MV411 xenografts. The animals were dosed 3 times. This is followed by sacrificing 3 mice at a time, an hour later, 2 hours later, 4 hours later, etc. And we can see the degradation of the BRID4 compared to the vehicle control, and that's illustrated here. We can see at 9 and especially at 10 and 12-hour time point, there's significant degradation, close to 25% is left. And after 24 and 48 hours, we see the rebound, which guides us to what we would be using for a dosing schedule. So just would like to summarize then, the cure pros provide the first reversible self-assembling protein degraders. And what they do is they combine the best features of cofferons, which are small molecules, and PROTACs, which bring together bivalent molecules to generate small molecules that reversibly assemble inside the cell to guide protein degradation through the natural recycling machinery. Cure pros are modular in nature, and this allows for combinatorial screening, resulting in rapid and cost-effective identification of the best lead compounds. The combinatorial platform significantly reduces synthesis time and effort to identify the optimal linker orientation at length and the E3 ligase partner, in other words, cereblon, VHL, MDM2 to each of the targets. We have demonstrated in vitro proof of efficacy of dozens of cure-pro pairs using several different bioorthogonal reversible linker chemistries, and this is very dependent on the geometry of the linkers. The mechanism of action is through proteasomal degradation, and further good in vivo absorption and exposure and the tolerability has been demonstrated. And finally, the in vivo proof of mechanism of lead cure-pro pair in a mouse xenograft model. I'd really like to close and thank my wonderful students, postdocs, and collaborators. The majority of the cofferon and cure-pro work was done by Sarah Giardina. More recently in my lab, Elena Valdebrini and Manny Bacalode. Also, original cofferon work by Manish Pingley and Lee Arnold. More recently, Ganesh Babu, Mike Peel, and Uta Warner in guiding the cure-pro work. David Warren at Weill Cornell, as well as Weijin Gui and Tom Kodadek at UF Scripps Biomedical Research. Thank you so much.